Today's video is about cytogenetics, or our ability to look at chromosomes and obtain important information from them. Shown here is a metaphase chromosome spread. This is the set of all of the chromosomes that were in the nucleus of a cell at metaphase. They appear blue because these chromosomes have been treated with a dye called DAPI, which binds to nucleic acid and lets us see where the chromosomes are. One of the things that cytogeneticists, who are geneticists that study chromosomes, can do with a metaphase chromosome spread is build a karyogram. And a karyogram is what you call an image where you've taken all of the chromosomes from a metaphase chromosome spread and arranged them so that the two homologous chromosomes are next to each other and in the same orientation. In this human karyogram, for example, you see that we are diploid. We have two copies of every chromosome. There are two copies of chromosome 1, two copies of chromosome 2, and so forth, until we get down to the smallest chromosomes, 21, 22, and the X and the Y chromosomes, the 23rd pair. Thus, this is a male karyogram. There's another closely related term that we use, a karyotype. And a karyotype is simply a text description of what we learn from the karyogram. So the karyogram displays, in this case, 46 chromosomes, and this is an XY or male. The last important piece of terminology here is the idiogram, which is different from a karyogram in that this is a representation of the chromosomes but based on drawings or cartoons of the chromosomes, not actual micrographs or photographs taken underneath a microscope. So a micrograph is used to produce a karyogram. An idiogram is just based on a cartoon. To construct a karyogram, you start with an image of a metaphase chromosome spread as it exists, and then you digitally, these days, cut out each individual chromosome, rotate them so that they're all in the same orientation, like has been done in this karyogram, and then line them up in a few different ways. Here the chromosomes are arranged first based on their centricity, and within each class they're arranged from largest to smallest. The metacentric chromosomes are chromosomes where the centromere, or primary constriction, is roughly halfway between one pair of telomeres and the other. So metacentric means middle centromere. In a sub-metacentric chromosome, the centromere is offset from the middle, but is not very close to the end as it is in an acrocentric chromosome. In the acrocentric chromosomes, you can see a tiny little bit of nucleic acid above the centromere on each chromosome. This is the main distinction between an acrocentric chromosome and a telocentric chromosome, where it appears that the centromere and the telomere are essentially the same object. There is no short chromosome arm as there are in submetacentric and acrocentric chromosomes. And because I just mentioned that, we're going to talk a second about chromosome nomenclature in terms of the arms. On either side of the centromere, we have for submetacentric and acrocentric chromosomes, the short arm and the long arm. There's the long arm, there's a short arm. Cytogeneticists refer to these as P and Q arms. P supposedly stands for petite or short, and Q for the word queue or long, like standing in a long line or a long queue. And so, in sum, that's how you construct a karyogram. You cut out digitally the image of each chromosome, orient them in a similar fashion, and then arrange them into metacentric, acrocentric, telocentric, and submetacentric groups, and arrange them in size from largest to smallest. Now, hopefully this has raised a couple of questions in your mind about how cytogeneticists actually go about doing this. The first concern might be regarding metacentric chromosomes. If both of the arms are the same size because the centromere is in the middle, then how do we know that this arm is the same as that arm? Maybe we don't know. Maybe these two arms of the chromosome are the ones that are related to each other. It's hard to tell just by looking. So that's the orientation of metacentric chromosomes. Maybe one of these chromosomes is meant to be flipped in this orientation to line up properly with its homologous chromosome, the one next to it. And a related question is, are we really convinced, for example, looking at these chromosomes here? So for these chromosomes, for example, how do we know, how sure are we that those two chromosomes are really the ones that are supposed to be paired up? They're the homologous chromosomes. Could I maybe convince you that this chromosome actually is paired with that one, and this one with that one? It's hard to tell when the chromosomes are the same size or almost the same size, and the same or almost the same shape. So that's the problem of identification of homologous pairs of similarly sized and shaped chromosomes. Fortunately, we have a technique that we can use to deal with these problems. And this is a technique called fluorescence in situ hybridization, which is the acronym FISH. And to perform FISH, we need a couple of things. One thing we need is a probe. And a probe is, as it says here, a piece of DNA that we have artificially made fluorescent. So in this case, I'm going to diagram this probe as a red fluorescent piece of DNA. And initially, it's a double-stranded piece of DNA. So I'm going to highlight this in red because it's presumably a molecule that emits red fluorescence. Then we also need a genome, the chromosomes that we're interested in studying. So you need a metaphase chromosome spread. I'll just depict a pair of metacentric and a pair of submetacentric chromosomes, like so. And ultimately, the result of doing a FISH experiment is that you find out where in the genome this probe sequence exists. And the results of FISH experiments look somewhat like this. There will be dots of that red fluorescent signal that sit on the chromosomes of the metaphase chromosome spread where this DNA sequence, whatever it is located. So in this case, this tells us that it's the long arms or Q arms of this submetacentric chromosome where this particular DNA sequence is found in the genome. Now if I switch to a different color probe, let's say we also have a different DNA sequence that we've fluorescently labeled blue, we can illustrate how this helps us solve some of our problems. For example, for the metacentric chromosomes, if that blue probe hybridizes to that metaphase chromosome there, and to this arm on this chromosome, now we know which arms of the chromosome are related to each other. those arms. So if we drew the karyogram, we would make sure that those arms that contain the probe sequences were paired up next to each other like this when we produce the karyogram. So this is what a fish experiment looks like in reality. We have a metaphase chromosome spread with one of the chromosomes sitting over here. And we have a DNA sequence, don't know what it is right now, but that's not really important, double-stranded DNA sequence, and that has been applied to this chromosome spread, and it's highlighting for us where in the genome exactly, on which pair of chromosomes is that sequence found. So now we know that these two are homologs. And so hopefully you can see this makes creating karyograms a lot easier. If you have one probe for every chromosome, then you can do a fluorescence in situ hybridization or fish experiment and tell out of all of these chromosomes that basically look like they're almost the same shape and size, which ones are the homologous pairs. Now here's where fish really works. When I draw a metaphase chromosome like this, let's actually imagine that what it really is two molecules of double-stranded DNA that are closely opposed at their centromere. Sometimes this looks like the chromosomes are overlapping, and sometimes they are, but they're not necessarily overlapping. The first step in a fish experiment is to heat or to denature the chromosomes so that they become temporarily single-stranded. So the hydrogen bonds that are holding the double-stranded molecule break. And this is where our DNA probe is useful. You also heat up to denature that short, originally single-stranded molecule, but when it's heated, it also becomes single-stranded temporarily. Now if I add some letters here, let's use a dinucleotide repeat. Say that this molecule has the sequence AT, AT, AT. And let's make up a short sequence of nucleotides for the chromosome that we're looking at. Now the next step is, while all of these molecules are together, you mix the probe with the chromosomes, you slowly cool the mixture back down. And what happens then is that single strand with its sequence, the chromosome that's affixed to a glass slide, is sitting there with all of its hydrogen bonds unsatisfied. And along comes one of these probe sequences that has a complementary sequence to part of the chromosome sequence. And so it will base pair with the chromosome. And now you have a blue fluorescent molecule that's giving off blue light when we look at it under the microscope. And that's how we actually visualize where in the genome that probe sequence is found. Now, clinical cytogeneticists use fish techniques and other techniques to identify 4 different types of issues that people can face with their chromosomes: deletions of DNA, duplications of DNA, inversions between chromosomes, and translocations between or within chromosomes. So we'll look at one example of each. Here's what a deletion might look like. So far, this looks like a pretty typical metaphase chromosome spread. The chromosomes are shown in white, and there's one fish probe that has hybridized to 2 spots in the genome. And that's standard for a diploid organism, which this is, like humans. There's 2 copies of every chromosome. So we expect to see 2 chromosomes exhibiting signals for a unique fish probe like this one. But you'll notice that if I diagram the chromosomes like this, on one of the chromosomes, the one in the upper half of the figure, the fish signal, which I'm going to denote as a black asterisk, is at the end of the chromosome. On the other chromosome, where there's a fish signal, it's in the middle. One of the things that this might mean is that there used to be some DNA sequence here on this first chromosome that has recently been deleted, leaving that fish signal apparently on the end of the chromosome, where on the homologous chromosome it's not on the end, it's in the middle. So the change in location on a chromosome of a fish probe can be indicative of the presence of a deletion of part of the chromosome. And as I've mentioned, it is possible to have multiple fish probes that have 2 different sequences. So we could have sequence 2 with a red fluorescent molecule and sequence 1 with a blue fluorescence molecule. These sorts of experiments, looking for duplications and inversions, can really benefit from using multiple colors. So in this example, what these scientists have done is they've got 2 different unique fish probes. Here they're green and red. And they're looking at a chromosome 15 from one individual and from a second individual. And you'll notice that in individual 1, there's 1 red signal, meaning there's 1 copy of DNA sequence C251-3, and there's 1 pair of green signals, indicating that there's 1 copy of T216-1. But in this other individual, there are 2 sets of red and green signals, which suggests a duplication. There's the original version, and there's been a copy. Detecting inversions also benefits a lot from having multiple colors of fish probes. In this case, I'm going to diagram the 2 chromosomes that exhibit fish signals. So this chromosome in the lower half of the panel is the one that I'm going to designate chromosome A. So I'm going to call that chromosome A, and I'm going to call this one chromosome B. And we can already tell that they're a little bit different, because despite having the 2 fish signals, which suggests that these are homologous chromosomes, they have different shapes. Chromosome A looks submetacentric. Chromosome B looks metacentric. In this case, the white dots that I've drawn on this panel represent the location of the centromere. Now in this experiment, we have 3 different probes, so I'll call them probe 1, probe 2, and probe 3. And because we only have 2 colors but 3 probes, 2 of the probes necessarily are going to be the same color. So we're going to say probes 1 and 2 are green, and probe 3 is purple. If we look at the location of these probes on chromosome A, it looks like the purple signals are above the centromere, and then there are 2 pairs of green signals, one near the centromere and one about halfway down the long arm. If we look at the location of signals on chromosome B, it looks like there's a fish signal just above the centromere. There's another pair of green signals down near the telomere, and in between the green signals there's a purple signal. The bottom line is the difference in the order of signals, purple, green, green on chromosome A, but green, purple, green on chromosome B, means that although these are homologous chromosomes, the physical structures of the chromosomes aren't identical. There's been an inversion in this case. And what this likely means in this instance is that what used to be chromosome A broke here, where I'm showing this red line, and flipped upside down, and then reconnected to produce a chromosome that looks like chromosome B. In sum, if you have 3 probes and their order appears to change on multiple chromosomes, that can be indicative of inversions. Last, we can look at how fish experiments help us identify translocations. In this case, instead of showing an entire metaphase chromosome spread, I'm only showing the 3 chromosomes that hybridized to fish probes. In this case, we have one chromosome with a green signal near a telomere, another chromosome that has a green signal near a telomere, and then the chromosome that has purple and purple. So when we look at this metaphase chromosome spread, there's something strange going on. Normally we see pairs of chromosomes that have the same fish signal hybridization patterns. But in this case, we still do have diploidy, or 2 copies of every chromosome. You'll see we've got 2 chromosomes that hybridize to the green probe, and we also have 2 chromosomes that hybridize to the purple probe. The strange thing is that one chromosome demonstrates hybridization to both the green and the purple probes. So what's the interpretation of this? This suggests the presence of a translocation, that is, the physical fusion, say, of the telomeres of this chromosome to the telomeres of that chromosome. And if you add those 2 chromosomes together, like that, you get something that looks like the 3rd chromosome. So the way this is strange, again, to summarize, is that we have a diploid organism, but it has 3 chromosomes that hybridize to the probe, not an even number of chromosomes. In a diploid individual, every chromosome should be present in 2 copies. So you should always see 2 chromosomes hybridize to a fish probe. In this case, though, there's an odd number, and that suggests the potential for a translocation to have occurred.